Welcome everyone, welcome Janelia and welcome EMBL to the very first bioimaging seminar series of 2022. It's my pleasure to welcome all of you to the new start of the series this year, and I would also like to welcome our first speaker, Karel Svoboda. So it's my pleasure to welcome you to our seminar series, and just a brief introduction before we dive into his presentation. So Karel was actually a group leader here at Janelia until December 2021. And he's right now the Vice President and Executive Director of the Allen Institute for Neuronal Dynamics in Seattle, Washington. At Janelia, Karel's lab was working on the structure, function, and plasticity of neocortical circuits, mostly in the context of somatosensation. He has a long-standing interest in the development of optical, physiological, and molecular methods for neuroscience. He received a bachelor's in physics from Cornell University and a PhD in biophysics from Harvard University. And before joining Janelia in 2006, he was actually a professor at the Cold Spring Harbor Laboratory and an HHMI investigator. In today's talk, Karel will talk about the past, present, and future of calcium imaging to spy on neurons in their native habitat. So it's my great pleasure to have you right now with us. Thank you very much for making it, because I know you're right now in Seattle and it's 7 a.m. where you're located. So thank you very much again. And yeah, please feel free to share your screen and please take it away.
All right, great. Okay, so good morning, everybody. It's really great to be here. Ulrike invited me to give a talk at this nice series, probably now close to a year ago, and I was a little reluctant because I haven't done a huge amount in my interest in optics waxes and wanes, and haven't really done much of great interest recently, but I thought it might be fun to kind of take a look back at the field of calcium imaging to track neuronal populations and ask questions about how did calcium imaging become this amazing tool for tracking neuronal activity, or actually population activity over the last couple of decades. What were some of the key inventions? What was luck? What was intentional engineering? How did kind of different technologies come to bear on the problem, and so on and so forth. And so just to set the stage, this is the kind of a problem that systems neuroscience tries to address. The brain is, unassuming on the outside, perhaps looking not much different than your liver and about, just a little bit larger. But underneath is great complexity. The human brain, for example, consists of 100 billion neurons. The mouse brain, which luckily all mammalian brains share cell types and rough organization, still contains 100 million neurons. Neurons are diverse. They signal through electrical signals called action potentials, which are depicted here as flashes of light, and neurons are connected. When one neuron fires an action potential, this millisecond timescale electrical signal, it makes action potentials more or less likely in connected neurons, right, through excitatory inhibitory connections. And this is how electrical signals propagate through circuits, and what underlies computation and ultimately behavior, okay? So wouldn't it be great if we could image the electrical activity of large neuronal populations, right? These signals that underlie our thoughts and our actions, our memories, our fears, and so on and so forth, using modern microscopy approaches, right? Wouldn't it be great if we could learn about these fleeting patterns of signals which underlie really all mental processes? And remarkably, this is now possible with calcium imaging. And calcium imaging is, of course, is only indirectly related to voltage imaging, to these action potentials, right? And so how did it, and this is now by far the most widely used method for tracking neural activity, with thousands of papers published each year. So on the left, you see a nice example from Janelia from Misha Aron's lab, a collaboration with Philip Keller, where a protein indicator was expressed in virtually all neurons in larval zebrafish. And the flashes of light here correspond to individual neurons lighting up with action potential, small trains of action potentials. And these trains of action potential increase the calcium in neurons, which then changes the properties of the indicator, which can be visualized here with a light sheet microscopy. Now, on the right is an image of multiple brain regions of the mouse brain. This is sort of looking into the mouse cortex in a behaving mouse. And again, you see these flashes of light that correspond to these dots here, individual neurons that correspond to the activity of individual neurons, maybe small trains of action potentials. So what are the key advances over the last 3 decades that made these kinds of experiments possible? I should say that the overall vision of imaging activity in populations of neurons. So if the classic method, of course, is electrophysiology, and that is also undergoing a revolution. Electrical measurements and optical measurements are kind of beginning to share properties. That's perhaps the subject for another talk. But the vision of imaging activity in populations of neurons has been part of neuroscience for a long time. This is actually from a review paper from the 1980s, really pioneering work from Larry Cohen's lab and Aram Greenwald's lab, who screened membrane dyes that had, and Leslie Lowe as well, that changed their fluorescence properties, okay, that intercalated into membranes. Of course, voltage has to be sensed in the neuronal membrane that would intercalate with these into the membrane and would change the fluorescent properties or absorbance properties with electrical field changes through the so-called Stark effects. And they were able to find dyes, for example, dye 8 ANEPs and others that you can put onto neurons that then would show small voltage changes. Okay, so on the order here, note the scale bar on the order of 10 to the minus 5 and 10 to the minus 4. So, hundredth of a percent changes in the properties. Nevertheless, in large neurons, often in the ganglia from invertebrates like aplysia, they were able to detect single action potentials and even record the activity of dozens of neurons here in the aplysia buccal ganglia. So this is pioneering work. But one question perhaps is, for the purposes, right, of recording large populations of neurons, why has sort of calcium imaging galloped ahead? What are some of the key advances? That's really what I'd like to address today, and to also give you a sense of how kind of different insights and technologies interplay in moving the field forward. By the way, another topic, of course, that deserves your attention is great advances in voltage imaging recently, also through the work of, among others, of Eric Schreider and Luke Levis and others at Janelia. All right, so here's the menu for the next 40 minutes or so. I'd like to tell you a little bit about the basics of calcium imaging, calcium signaling going way back. I'd like to remind you of the classic era of calcium imaging, which really laid the foundation conceptually for tracking neural activity using calcium imaging. Again, familiar to many of you and critical for the development were microscopes that allow high resolution, high contrast imaging in vivo. And then a very intentional step at Janelia over the last decade, the engineering of protein calcium sensors that made calcium imaging much more practical in interesting biological situations, and which I would argue was one of the key events for unleashing the technology to many laboratories, making it straightforward, turnkey, and amenable to many kinds of applications. And the development of high sensitivity protein indicators then again led to a renaissance, a second renaissance in microscopy methods, which is continuing to press ahead. All right, let's start with calcium imaging. And as I mentioned, this is a retrospective talk, so let's start very much at the beginning. And interestingly, we of course know that the Earth crust is about 4% calcium in the form of limestone, but also gypsum and fluoride. But until recently, we didn't know where that calcium came from because most observed galactic event with thermonuclear fusion actually produced very little calcium. It's a little puzzling. Until recently, folks discovered that certain kinds of white dwarfs had the right kind of condition for thermonuclear fusion that actually produce very high levels of calcium, which then radiates through the universe and is likely the source of calcium on Earth. So even the very, very early aspects of creation of calcium is still the subject of research in astrophysics. Now on Earth, of course, and in biology, calcium gives rigidity to bones, as you know, but for the purposes of this talk, we've known for close to 40 years that calcium is a ubiquitous second messenger. And what that really means is that it's a signal downstream of extracellular signals in most cells. And what folks think is sort of the standard story is that early cells in the oceans had to learn how to keep intracellular calcium low, likely to avoid the precipitation of phosphates, which are of course critical in energy metabolism. So the first event in calcium signaling during evolution was figure out how to extrude calcium from cells to keep calcium low in seawater, but calcium, of course, is very high on the order of 10 millimolar. And now in most situations, the intracellular calcium is low on the order of 100 nanomolar, 10,000 times lower, or maybe even 100,000 times lower in ocean microbes than extracellular calcium. Okay, so cells have developed specialized mechanisms very powerful mechanisms and energetically costly mechanisms to extrude calcium. And importantly for this talk, cytoplasmic calcium routinely changes 10 to 100-fold during physiological signaling. So 100 nanomolar on the inside, a millimolar on the outside, so strong driving forces for calcium across the membrane, and during normal signaling, calcium changes often 10 to 100-fold. Now, of course, unlike many other second messengers like IP3 or cyclic AMP, calcium cannot be chemically altered. So to exert control over calcium, cells have efficient mechanisms to extrude calcium in neurons. That's calcium ATPases and the sodium calcium exchanger that are remarkably efficient, as I'll mention in a second. And other ways to control calcium is to bind calcium. So there are literally hundreds of proteins in the genome that bind calcium over a millionfold of range of activities. It turns out that different compartments in the cell signal with calcium over different ranges of concentrations. Some of these proteins buffer calcium and keep calcium dynamics very brief, okay, for signaling. This is like paravalbumin and calbindin, or even lipid membranes. whereas other proteins like pumps lower calcium or trigger cellular processes. And it turns out that calcium binding to proteins is one of the key events in signaling, changes the electrostatics of proteins, which then can cause, of course, large conformational changes in calcium that initiates signaling. And a canonical example here is calmodulin, which is also most often used in creating protein sensors for calcium that with calcium binding undergoes a large conformational change to initiate signaling, here by binding around its, by wrapping itself, binding to and wrapping itself around its target peptide here, for example, of a kinase. Now, how about what's special about neuronal calcium signaling? So again, over the last 3 decades, we've learned a great amount about calcium signaling. We know most of the sources of cytoplasmic calcium, the sources that change the second messenger concentrations. This includes synaptic inotropic receptors. So at synapses, neurotransmitters bind these inotropic receptors, open these receptors, and a subset of these receptors are highly permeant to calcium, in particular, for example, the NMDA receptor, which is a critical mediator of linking neural activity to calcium influx, which then changes synaptic strength that underlies learning. The certain acetylcholine channels are membrane permeable, and certain types of AMPA receptors are calcium permeable. But calcium also enters cells through the activation of G protein coupled receptors, which then couple through other second messengers, perhaps through the release of stores. And we know quite a bit about what these calcium events, the calcium that comes through these different pathways might do, like modulating synaptic plasticity, or in some cases, mediating the transcriptional responses of neurons. But it turns out that the vast majority of calcium coming into neurons during their normal function is associated with action potentials through voltage-gated or voltage-sensitive calcium channels. And remarkably, even though that amounts to perhaps 99% of the calcium coming into cells during the normal function and is relatively global in neurons, as I'll show you, we know relatively little about the function of this massive and frequent calcium influx into cells, into neurons. Nevertheless, it's this influx of these voltage-gated calcium channels during action potentials that is imaged in these movies that I shown you earlier. Now, an important also insight was that neural signaling is fast. Okay, so and this again can be looked at with calcium coming in response to action potentials. So action potentials, for the purposes of our discussion, essentially delta functions in time. They're on the order of a millisecond, and they cause these rapid tail currents. Okay, they activate voltage-gated, voltage-sensitive calcium channels, and then on the downward swing of the action potential, you see this blip of calcium current, again on the order of a millisecond in a duration, again a delta function. And when one looks at calcium with quantitative methods, these are imaging methods that I have not yet introduced, we find that in many neuron types, the decay time of the free cytoplasmic calcium transient is on the order of 10 to tenths of milliseconds, perhaps 100 milliseconds in very large neurons. It actually depends on the surface to volume ratio of the neurons and on the exact nature of the extrusion mechanism. This really reflects the fact that neurons really like to get the calcium out of the cytoplasm quickly. So the residence time of calcium ions in the cytoplasm is on the order of tenths of milliseconds. So what that means is that the dynamics of the cytoplasmic calcium, which you actually image in calcium imaging experiment, has the kind of similar dynamics to the dynamics of action potential that allows us to, in principle, image the dynamics of action, extract the dynamics of action potentials from the dynamics of calcium. So this is conceptually what we're doing, right? By measuring calcium in neurons, we can extract electrical activity to first order, okay? And this is really like measuring perhaps the timing of lightning based on thunder. It's a bit indirect, but really pretty good for most purposes. So am I doing on time? I'm really a little slow here. So let's talk a little bit about the classic era of calcium imaging. And this is not really an imaging experiment, but it's, I think it's really cool. And it's as far as I know, the first optical measurement of neuronal calcium. And this was done with the recently discovered aquarin from Aquaria Victoria. In fact, right now, if you look out of my window, I'm overlooking the Puget Sound, okay, where Aquaria Victoria was first collected by in Friday Harbor up there, and aquarin was isolated, and aquarin complexes choline, okay, and calcium coming into here, the squid giant axon that was injected with aquarin that was collected in long summers by chemical experiments in long summers was injected with aquarin that is complex with the cofactor choylentrazine. I think it always gives me trouble, choylentrazine. And calcium coming into here, the squid giant axon in response to trains of action potentials then catalyzes the oxidation of the choylentrazine, which leads to the emission of photons, which can be detected with photomultiply tubes. And you can see increase in calcium level with trains of action potentials. This is really the first direct demonstration of calcium coming in from the outside to the inside into neurons associated with action potential. So of course, this is slow. This is an unbelievable tour de force. 2 tons of crystal jellies yield about 100 milligrams of aquarin, enough for a few examples. So this is not a method that has been used a lot, but it really set the stage for what came later. Later, chemists came into the field, and there's a long history of interesting but somewhat archival work that I'm skipping over that is worth review, perhaps by a chemist at some point. But the major breakthrough in calcium imaging came from the laboratory of Roger Chen, who later on, of course, played a great role also in the engineering of green fluorescent protein. And Roger's lab really set the field on fire by developing a series of reagents to manipulate intracellular calcium and to measure intracellular calcium by coupling calcium buffers to fluorescence changes in small molecules. And these are some of the key reagents. These are calcium buffers here that in particular BAPTA, these oxygens here can complex calcium, and BAPTA was revolutionary because it's a calcium buffer that you can inject into cells, interfere with calcium signaling, establish causality between calcium dynamics and signal transduction events. It has much nicer properties than the previous EGTA. In particular, it's not pH sensitive and relatively inert, and so on and so forth. Now, BAPTA became the basis for famous calcium sensors like Fura2. Here's BAPTA and the chromophore Fura2 that changes its spectral properties with calcium binding, allows ratiometric analysis. And here, BAPTA associated with xanthine dye, where calcium binding in flow 3, that is the name of this molecule, can change the fluorophore brightness by 40 to 50 fold. Okay, huge signal to noise ratio. And these kinds of indicators became the basis of a lot of what we know about the basics of calcium signaling in neurons and have been and in other cells. So as these indicators came around, other advances then allowed in fluorescence imaging, in particular the development of sensitive sensors. This was in this era called CCDs, which made their way into biology from astronomy. Okay, they were developed for astronomy. The price point was sort of in the early experiments not too different than a true photon microscope, hundreds of thousands of dollars. But these things, these cameras and these calcium sensors made these kinds of experiments possible, where folks began to study the spatial temporal dynamics of calcium during physiological activation of neurons. So here you have a brain slice and the image of a neuron. The image of the neuron in this epifluorescence microscope is hazy because of scattering. Of course, in the brain slice, the neuron is perhaps 100 microns in a highly scattering tissue. That's why this neuron sort of blooms out. The cell body might be here, and there's this haze by its dendrites. Okay, this is now the calcium in pseudo color under resting conditions, and then with its synapses activated. Okay, great increases in calcium, in particular in the dendrites, which then come back to resting conditions. So these, 100 nanomolar regime, and then rise up to close to millimolar in the dendrites. Later, it was shown that this calcium increase in the dendrites is due to action potentials that radiate out from the soma into the dendrites. And there are also some interesting artifacts in these images that were later understood. So, for example, the calcium indicator was infused into the cell through a micropipette right at the soma, and there's a very large concentration of calcium indicator in the soma, and the calcium indicator acts like a buffer which reduces the free calcium change at the soma, where the action potential has the largest amplitude. And so there's this sense here that there's low calcium at the soma, which is not necessarily true. It's actually lower because of signal-to-noise ratio, but not to this extent. This is because the calcium indicator concentration is very, very high, because that's where the large source of calcium buffer is from the electrode that you don't see here. Now, so we learned a lot about calcium using these measurements, and it's maybe worth asking a conceptual question, and I think you know the answer to this: why imaging calcium as opposed to the many other ions that change to a larger extent in absolute terms than calcium, like, for example, sodium. There's lots of course, the main depolarization of the action potential is due to a sodium conductance, which admits a lot of sodium ions into the cell, or potassium that causes the downswing of the action potential, right? Why not image sodium and potassium? And the answer is simply, I gave you the answer already, is that only calcium changes, on the log scale, multiple fold, increases from resting several fold during normal signaling. In contrast, even though sodium might change by several micromolar, perhaps tens of micromolar, because sodium concentration inside the cell is on the order of several millimolar, the fractional concentration change is only on the order of 10 to the minus 4 compared to 10 for calcium, and similar considerations apply for potassium and chloride. So even though very good synthetic indicators, again, through the work of Roger Chen and others, became available, the signal changes are very small because these molecules work with first order binding. You move very little here related to, on the binding curves. So you have very small changes in the fluorescence change during normal signaling. Here's another important early experiment that told us that you can look at action potentials with calcium imaging at reasonable firing rates, again, because of the fast dynamics of calcium. This was done with xanthine-based indicator in brain slices. Okay, so for imaging populations of neurons, we have to improve imaging, right? So here, this spatial scale is 100 milliseconds. You know, with the scattering, this blurring of scattering, we would mix up many neurons in the field of this dendritic tree. We need much better spatial resolution in intact tissues to read out the population of to read out neural signals using calcium imaging from populations of neurons. So a major advance were microscopes for high resolution imaging in intact scattering tissue, in particular in vivo, and ultimately, of course, the dynamics of neurons have to be probed in intact brains. And here, some of the conceptual advances go back to the early days of quantum mechanics. Read the work of Gerbert Meyer, who was a student in the Göttingen School of Quantum Mechanics. And in her PhD thesis, she had the task of looking at multi-photon absorption in atoms. And she showed that the absorption rate depends on the second power of the incidence and intensity. This is really, you can think of it as you need 2A to make an excited B, you know, so this reaction is dependent on the second power of the reactant. And Gerbert Meyer already noticed in her thesis that very, very high light intensities would be required to observe two-photon excitation. And only at the time of her death in the 1960s, she died relatively young. I think it was in the maybe 1970s, but around the time of her death was two-photon excitation observed that, of course, demanded the invention of the laser that could produce very high light intensities, and then ultimately was practical for microscopy with the invention of mode-locked pulse lasers, which actually produce peak intensities that are larger than any in the known universe. And she later actually received the Nobel Prize for another fantastic piece of work on the structure of the nucleus. And she did that work without having an academic job in the US. So really quite remarkable scientist. Nevertheless, this is a nonlinear effect and underlies most of the advantages of two-photon excitation microscopy for imaging in scattering tissues. This is well known. I won't belabor this. I think many of you know the principle. Suffice it to say that even in scattering tissues, two-photon excitation provides the nonlinearity of two-photon excitation provides localization of excitation. So excitation is limited to the tiny diffraction-limited laser focus. Because scattered photons are really too dilute to produce off-axis, off-focus excitation to first order. Okay, and then because all of the fluorescence light comes out of the excited diffraction-limited volume, it can be collected as useful fluorescence rather than producing background or and so on and so forth, like it would in confocal microscopy. So the proof is in the pudding. So rather than these kinds of images, and by the way, this is work that really Winfried Dank in the middle 1990s figured out why two-photon excitation is particularly useful for imaging in scattering tissues. When you read the original two-photon microscopy paper from 1989, there's no mention of this application. This work really was done at Bell Labs, and really Winfried Dank really did the kind of the key work and figure out the key underlying concepts. In hindsight, simple, but quite surprising at the time. So these are the kinds of images we're able to acquire with early two-photon microscopes. Again, we labeled cells in this kind of painstaking, backbreaking way with microelectrodes where the microelectrode introduced fluorophores is a Purkinje cell. You can see this exquisite, is in a brain slice, exquisite resolution, individual dendritic spines that are on the order of 0.5 micrometer become resolvable. And this even works in vivo. So this is now in an intact rat brain. Here's the surface of the cortex. Again, the electrode that penetrates the neuron, now introducing a synthetic calcium indicator. And here we were able to show that in the intact brain, calcium changes are rapid as advertised because of the very short calcium current associated with action potential. So with the electrode, of course, we can do electrophysiology, and the fluorescence decays rapidly because of the powerful extrusion mechanism, actually slowed here somewhat because of the presence of the calcium buffer, one of Roger Chen's dyes, or one of the successors of Roger Chen's dyes. But you can already see that by looking at the calcium-dependent fluorescence in this neuron, one can extract a lot. One can learn a lot about the underlying neural activity. Now, introducing fluorescent substances into living neurons in the intact brain is inefficient. Okay, and so labeling nervous systems in an efficient, non-destructive manner really became a huge technical challenge. And a step was made, in particular, by Arthur Conrath and some lovely papers in Clay Reed's lab, by using AM esters, again, chemical indicators, or the dyes that Roger Chen made. He made also a bunch of ester versions that are membrane permeable, that are then trapped in the cell through the action of esterases, but and leading to lovely studies, for example, looking at the organization of direction or orientation tuned neurons in here the cat visual cortex. But this was still a very challenging method. It required chemical insults, DMSO, to dissolve the esters. So it was effectively a one-shot experiment that lasted on the order of an hour or two and was destructive to the tissue. And so not easily compatible with the long-term experiments that we'd like to do in behaving animals routinely, or experiments doing behavior where one invests in training experiments. So this challenge of getting indicators into cells in a non-destructive manner was addressed with protein calcium sensors. So I'll talk a little bit about engineering at protein calcium sensors, and really at the of course the seismic event that came as a complete surprise to many of us, sort of the bombshell in the mid-90s, was the demonstration that green fluorescent protein expressed in cells and even in intact animals by itself produced a functional fluorescent molecule. And with a little bit of engineering, it turned out to be a great fluorescent molecule at that, with high quantum efficiency, good brightness, little cytotoxicity, excellent to photon cross-section, okay? And then with a bit of engineering, spectral and bioprospecting, spectral mutations were found, of course, over a lot of the visible spectrum. Really a bit of a stroke of luck, I would say. And the invention of green fluorescent protein then led to a great era of protein engineering to generate functional probes based on green fluorescent protein for kinases, for cell death enzymes, and also for calcium very early in the game. So a number of groups that figured out how to make GFP calcium dependent in the late 90s, going in the 2000s. There were different groups, invented different scaffolds. These are just sort of two example scaffolds. There's a ratiometric concept here based on FRET, blue or cyan protein, yellow protein, conformational change using calmodulin that changes the distance between donor and acceptor, a well-known sort of motif. And another popular motif that that is a little bit better behaved and that sort of became dominant for calcium imaging is to use single fluorophore protein calcium sensors that are based on circularly permuted fluorescent proteins. Again, now with the new CNN terminus, calmodulin and M13 on the new CNN terminus, and then calcium changes the conformation of the fluorophore, changing its fluorescent properties. That's it in a nutshell, but for many years, the performance of these protein indicators just was hugely below that of synthetic indicators. They were really nearly useless for in vivo imaging, in particular in the mammalian brain. And these are some of the earliest Gcamps that we tested in the laboratory. And Gcamp has become the kind of dominant sensor in the field, originally developed by Junichi Nakai. And you can see that you see very little, even with 5 action potentials. The sensors were dim at mammalian temperatures. The protein was unstable. The signal changes were tiny, even with trains of action potentials. There was no way to extract the fluorescence from single action potentials, even in highly favorable conditions like in these brain slices. So at Janelia, we decided to pound on improving protein indicators of neural function. And there are at least 4 key parameters that need to be tuned. They need to be brighter, more stable at physiological temperatures, and more sensitive. The affinity of these indicators had to be shifted towards lower calcium concentration to overlap better with the normal dynamics of calcium in neurons. And we wanted sensors with faster kinetics. And so this engineering of sensors at Janelia was under the GENIE project. This is a project team now long in the tooth, about 10 years in existence. And the sole purpose of this group was for many years to develop protein calcium sensors and other sensors for neuronal function. And it's a group of scientists, protein engineers, outstanding technicians. I've also included some key alumni, especially folks in Lauren Luger's lab who made pioneering contributions over the years. And the basic concept is essentially screening and using methods of screening that are informative about the performance of the sensor in neurons that predict function in intact nervous systems. So early experiments in Lauren's lab revealed that Gcamp has multiple soft spots, places where mutations cause changes in function, often beneficial. Often these beneficial mutations are difficult to predict. And the more mutations are tested, the more the fluorescent sensors became improved, okay? And so the sensor pipeline really consists of a system to study thousands of sensors using quantitative biophysical assays in 96 well plates. So at the beginning is site-directed mutagenesis. These neurons are then transduced into neurons in 96 well plates and then studied in replicates. In imaging experiments, these images are then analyzed for function, for sensitivity, for kinetics, for baseline fluorescence, for brightness, whatever you'd like. And then the best hits are then analyzed in vivo systems. So a key aspect of this pipeline is testing, is an assay in primary neurons. and in 96 well plates. And neurons are critical because only neurons have this very fast calcium signals that these sensors need to pick up. And here is how some of these experiments look in the cultures. You can see that these Gcam sensors easily produce hundreds of % changes in fluorescence in response to trains of action potential. So we can produce electrical field stimuli that then produce action potentials in these neurons, look at fluorescence responses, measure the amplitude, the decay time of the fluorescence, and so on and so forth. And neural screening is worth it because the improvement of this is an outdated slide already, we should update it, note to self here. This is for example, the estimated signal to noise ratio. And as we learned how to screen more mutants, the improvements tracked the effort in screening. So screening is worth it. And that's because often it's difficult to predict the effects of any one mutation. So using these kinds of approaches, we were able to produce sensors that are now widely used in the field. Perhaps currently the most widely used sensor is Gcamp 6, now superseded by more modern sensors that are kicking in and becoming adopted. So let me turn this off. So this is Gcamp 6 here, a neuron that is also patched. Here's a patch electrode. These are action potentials and the fluorescence transients corresponding to the action potentials. So it, again, is close now with protein sensors, close to this dream by looking at calcium dynamics and being able to infer the dynamics of action potential, being able to infer action potentials, learning about the neural code in this neuron and its neighbor. This has been very successful. Thousands of labs are using these sensors currently. Briefly, how do these work? So again, Gcamp consists of a circularly permuted fluorescent protein. Here is the RS20 or M13 calmodulin binding peptide. This is the calcium bound form, the calcium free form, and see the calcium free form, the structure is incomplete. Nevertheless, you can see the large conformational changes of these helices with calcium binding. And the current hypothesis is that there is a hole in the beta barrel, in the circularly permuted beta barrel, that provides solvent access in the calcium free form, producing the proteination of the fluorophore tyrosine, and with calcium binding, that hole is plugged, producing a bright fluorophore. And importantly, again, here are some of the key mutations that led underlie, for example, the transformation from Gcamp 2 over 10 years to Gcamp 7, and it would be difficult to predict the sites of these mutations, even though a lot of them are related to sensitivity of the fluorophore to calcium. They're mainly at the interface between the beta barrel and the calmodulin. No beneficial mutations, I believe, and Eric can correct me here, I think he's on here, were ever found close to the EF hands, to the even kinetic mutations where calcium binding occurs. Kinetic mutations are really related to the interaction between the calmodulin binding peptide and calmodulin. In fact, Yang Zhang discovered recently very fast Gcamps, the Gcamp 8s, that make use of a completely different calmodulin binding peptide, because we were not able to increase the kinetics greatly with the RS20 peptide. So anyways, again, justifying the use of screening and high throughput assays to improve these sensors factor of 2 at a time. If you do this 3 or 4 times, you've improved the sensor by a factor of 10 or 20, and all of a sudden, very different kinds of experiments become possible. And I think this is an important lesson. Often we encountered criticisms, right? This, you're improving these things, in this kind of steady fashion where the breakthroughs, but you do a couple of these steps and all of a sudden completely new experiments and microscopy methods become possible. These kinds of improvements are key to expand the palette of use cases and experiments and the kinds of biological discoveries that can be made. Let me say a little bit about voltage imaging. Again, voltage imaging is also undergoing renaissance, but it's been challenged for this purpose of dense imaging of neural activity in the brain. And this is reason, and here are really the reasons. The voltage induced fluorescence changes for voltage sensors in the membrane have been limited. The dynamic range is sort of on the order of tens of percent, rather than for Geckies, where the dynamic range has been, thousands of percent routinely, often 10,000%. Then a huge disadvantage for voltage imaging is the fact that voltage imaging has to be sensed in the membrane, and 2D is simply small compared to 3D. The number of molecules per resolution element that one can stuff into membrane is relatively low than what you can stuff into a corresponding volume elements, producing a big signal to noise disadvantage. Also, voltage changes are fast, and calcium changes, depending on the off rate of the sensor, can be tuned, but can be slowed. And it's, of course, the integral under the response of the sensor that provides the signal to noise ratio, again, producing a signal to noise advantage for calcium imaging compared to membrane imaging. Finally, of course, voltage indicators are not perfectly trafficked to the membrane, and intracellular accumulations of the fluorophore produce non-productive background, reducing the signal to noise ratio. And so for these reasons, the signal to noise ratio for detecting neural activity has been much better for calcium imaging than for voltage imaging. But of course, voltage imaging is more direct, and for sparsely labeled populations of neurons, voltage imaging is providing a lot of interesting information that is invisible to calcium, in particular, subthreshold dynamics of voltage, and so on and so forth. Nevertheless, so these Gcamp sensors from the Genome Project and related reagents have enabled lots of studies. All plasmids are widely available, so are mice and flies at the Bloomington repo, mice at JAX, and that's where you can find this stuff. So finally, the amazing high signal to noise ratio of Gcam sensors has enabled new kinds of microscopy methods, and that's often underappreciated. And in particular, some of the Gcam sensors are essentially dark at rest, provide very little background, and can change their fluorescence by several-fold upon neural activity. And it's these kinds of it's these kinds of properties of the Gcam sensors that made these kinds of experiments, for example, selective plane illumination, possible, right? There's very little background in the absence of neural activity, which produces the high signal to noise ratio in these wide field imaging experiments. Selective plane illumination or even light field microscopy is now used in these transparent, relatively non-scattering specimens. Even wilder is people have started using standard wide field microscopy in vivo. This standard epifluorescence microscopy, again, enabled by the fact that these Gcams are essentially dark in the absence of neural activity, and light up when neurons fire trains of action potentials. By the way, these kinds of experiments also have been enabled by the development of new CMOS sensors based on the electronics revolution, smartphones, and so on and so forth. All right, so based on these new sensors and microscopy methods, really calcium imaging has become a very versatile method for probing the brain. I'm almost done here. And the kinds of applications I've shown you are sort of tracking activity or with dynamics on the order of tens to hundreds of milliseconds over hundreds of micrometers. But other applications are to look at synaptic dynamics over micrometers. Then one can track the same neurons over time during learning over timescales of months, looking at the properties of neurons over month. Again, very difficult to do with other methods. I want to just briefly mention, and I think Dan Flickinger gave a talk in this group recently, so I go over this rapidly, is to go here to the mesoscale, because neural activity in multiple brain regions, of course, underlies behavior, and multiple brain regions, even in the mouse brain, are separated over scales of millimeters. So we need to track individual neurons, but over scales of millimeters. And again, enabled by these new sensors that provide exquisite signal-to-noise ratio, we and others, and this is ongoing, endeavor to produce mesoscale microscopes that have subcellular diffraction-limited resolution, but over very large fields of view. And basically, the insight here is that even though in commercial microscopes, right, higher resolution microscopes have smaller fields of view, it's not a fundamental constraint, but really reflects engineering constraints. And for imaging calcium for particular applications, these constraints can be relaxed, and with new, perhaps more new methods of optical design, these constraints can be overcome. So we set out to make a microscope that can image single neurons over many brain regions. So here, for example, is the whisker somatosensory cortex, the somatic somatosensory cortex related to paws, and the motor cortex underlying whiskers and somatosensation. And it's these multiple brain regions that underlie motor controls. We wanted to image them all at the same time. So we wanted to have a microscope that has a field of view of 5 millimeters and still sub-micrometer lateral resolution rather than the typical 500 micrometer field of view. So we wanted a large microscope optimized for 2-photon, an excitation NA, numerical aperture of 0.6, a large working distance, and an emission numerical aperture on the order of 1 to collect fluorescence very, very efficiently. And I'm not going to, for the sake of time, won't talk much about the incarnation of this microscope, but just tell you that we've achieved this over a cylinder of 5 millimeter diameter, 1 millimeter depth. We have axial lateral resolution that is sub-micrometer, axial resolution on the order of 4 microns, that is sub-cellular resolution, and it really allows us to image large fields of view with subcellular resolution. This is now panning such a microscope. Now we can sort of zoom in to small regions, and you can see single neurons and their nuclei, these dark spots, this Gcamp, there's a Gcamp expressing brain. Neurons show up as these halos that, and here we're now focusing down, and these bright neurons are neurons that are active in this living brain going down 600-700 microns into the brain and zooming out. So allowing us to image now neural activity over multiple brain regions, for example. So we can scan a 5-6 millimeter area of the brain and select brain regions for high contrast, rapid imaging in this random access microscope. Okay, I'm done. I just want to summarize briefly what I've told you about. This is kind of the flow of kind of concepts, methods, discoveries that have led to the current state of calcium imaging for populations of neurons. This may be incomplete. I finished this morning, so if I've forgotten something, please let me know. I think really important is the imaging with small molecules sensors that showed that calcium can detect single spikes with high fidelity, and the underlying biophysics, that action potential of calcium is large, fast, and then it dominates neuronal calcium. Okay, this was made possible, of course, by inventions in electronic detectors and methods for high-resolution imaging in intact tissues. This really provided the impetus for looking at calcium as a high signal-to-noise ratio method for reading out population activity. At the same time, a separate thread in cell biology, molecular biology, with the invention of GFP, showed that GFP can be modified to produce excellent sensors for small molecules, including calcium. Together, this then provided motivation for doing protein engineering to optimize these sensors for in vivo applications. A very important thread I didn't talk about is efficient methods for gene transfer in vivo. And these new protein engineered sensors really made calcium imaging and all kinds of modalities, many of which I haven't talked about, easy, which expanded the field and also led and is leading and causing the next generation microscopy methods, which benefit from, again, new sensors, but also, again, having a chance to talk about new methods that democratize custom optical design. So last slide. So I think what are the trends? And none of these are surprising. You've probably heard presentations about many of these, and sometimes these predictions are wrong. There discoveries may come from unknown area of science. I remember an imaging symposium somewhere around 1993. I have some notes somewhere. The great luminaries were discussing about where imaging was going, and no one really predicted GFP or the power of a GFP, which in 1994 changed everything, right? So, but nevertheless, I think what's pretty clear is there is the return of real chemistry and chemigenetics, and really Janelia is leading, Luke and Eric are doing fantastic work, and Alison Thibault as well. There is a need for light sources and convenient light sources and fluorophores for ultra-long wavelength imaging that will allow us to go much deeper. There are interesting low absorption bands for excitation in the sort of 1.6 micron range and also 1.28 micron range. We need better fluorophores, not just for 2-photon excitation, but for 3-photon excitation microscopy, which is currently limited by the bleaching and absorption cross-section of fluorophores. I think a really exciting area that is very broad is computational microscopy to engineer microscopes and even excitation volumes, point-spread functions, but also perhaps fluorophores with image analysis algorithms in mind, and with deconvolution and AO, adaptive optics methods in mind. All right, sorry I went a little over. Thanks for your attention. Be happy to discuss this or any other thing that you might want to.
Yeah, so thank you very much. That was you gave an amazing overview. I mean, I myself, I'm not from the calcium imaging field, but I think I learned a lot over the last hour, and that was, as I said, it was very insightful. So, but since we're already over time, I would say we just give the audience the chance to ask maybe 2 more questions or so. So if there are questions, please raise your hand. Feel free to switch on the camera and also unmute yourself. Are there questions so far? Because otherwise I can maybe kick it off with also a question about the custom optical designs, which you mentioned for democratization also of calcium imaging. So I think on the one hand, I mean, I guess these dyes are already available via these platforms that you can also just buy the constructs and replicate this, but it's, I guess, different with the optical components and the hardware. Do you have something specific in mind how one can kind of optimize this, like specific platforms or some way to actually help other labs to take up the knowledge which is produced at Janelia or even at the Elm Institute?
Yeah, so there are many answers to this, right? So the first is I think so there's access to technology is something that, of course, we've taken very seriously at Janelia, and it's a challenge, right? It's a challenge for, it's easy for constructs. It's a challenge for, well, Ulrike, you know more about this than anyone, but I'll give you my perspective. So I think one way is, of course, what you're doing. There's this kind of barren spot, right, between, I think commercialization is really a great way ultimately to commercialize technologies. Not everything lends itself to commercialization, and there's of course a big gulf between a prototype in the laboratory and commercialization, because an instrument often needs $1 million or $2 million in R&D to make it in a turnkey instrument. So what we've done with the mesoscale microscope, and this was actually an idea of Jerry Rubin's, both for the lattice light sheet and for the mesoscale microscope, is to hold a workshop. And we had a workshop at roughly the same time that taught, in our case, about 14 laboratories how to build the microscope. So what they did is they would buy the custom optics and optomechanics and the 1 or 2 little pieces of custom electronics. And then over a 2-day workshop, we constructed a microscope. I have some great photos. I don't know if I have them here. This was really a lot of fun also. And literally a few of those folks had microscopes running a couple months later. And we also had Thor Labs people who were interested and hovering around, and they saw the interest and they worked through the processes themselves and then decided to license the technology. And they make, and you know, the market here is maybe a total of 50 instruments. So it's sort of great for a company like Thor Labs, but probably not for Zeiss, which took up the lattice light sheet, right? And some tools are smaller and have a smaller footprint, or might be even more complex, and may not be, for that reason, ever see commercialization. And I think as a tool developer, you have to be aware of this, right? I would shy away from, unless there's a real killer application, right? That's sort of, as a tool maker, you should think about right from the get-go, right? Dissemination should be part of the design process in some sense. The democratization of optical design, I had something else in mind, and that is that, and I'm actually a little bit out of my depth here, but the way I understand it is that computational methods have really changed optical design, and you don't need to be freak expert at Zeiss to do interesting things, right? You can design interesting things with Zmax. Of course, you need to have skills, and it's great to have a collaborator like Dan Flickinger, but you can also design, tolerancing, and you can use Monte Carlo methods to invent designs that are performant, but also relatively easy to build, right? And that's what tolerancing means, that none of the optical elements have to be aligned to 10 micrometers, but maybe only to a fraction of a millimeter. And that makes a huge difference. You can put cost of glasses into the design. And that's a big optimization problem, but kind of modern, standard large-scale computing kind of can get you interesting solutions that would have been hidden just 10, 20 years ago. Let's see, do I have anything else to say about that? Yeah, so that's really that's really it.
And maybe one last question, maybe from my side. I mean, we talked right now about the past, present, and the future, but what were career-wise maybe your, what was your highlight, or was there any kind of special moment during your career also in neuroscience where you had this feeling, oh my God, this is so amazing? I think you have this maybe all the time, but maybe one moment.
I've been lucky to have been sort of at the ground floor on a few things, and so I think I had this feeling in biophysics as a graduate student, but I'm not going to talk about that. Let me talk about. So at Bell Labs, when I went to Bell Labs to use 2-photon for a crazy experiment that only became possible 10 years later, again, because of the available chemistry. I won't describe that experiment, but it was when we realized that you could image with high resolution in scattering tissue, especially kind of the biology of neuronal microcompartments became accessible in a way that it hadn't been. And anything you looked at was new, right? So previously, folks had looked at synaptic transmission, hundreds of synapses at a time. You recorded the synaptic current at the cell body. Now you could look at synaptic dynamics using calcium imaging, for example, one synapse at a time, right? Like with single channel patch clamp that revealed a huge amount about how individual noise in individual synapses contributed to the fluctuations of the whole, signal and noise in synaptic transmission. Just anything you looked at became new. We could look at neurons over timescales of days, weeks, and months during learning, right? Again, was completely transformative and very conceptually very straightforward. So sometimes, you have this moment where a new tool gives you new vistas, and that's of course what new microscopies do. That's why we're all excited about new microscopes, new vistas, and tightly coupled to new biological insights. Then there was the appreciation that this was sort of technical, but it was very clear right away that calcium imaging would be big for tracking neural activity. Was the finding that really it's kind of surprising, because we don't know what calcium through voltage-gated calcium channels that comes into cells, right, with action potential, what it does. Nevertheless, it's kind of the dominant signal. And it's sort of the slop, perhaps, that allows us to do these large-scale imaging experiments. So I think that was cool. And then I was amazed by the early successes of protein engineering that initially were done in Lawrence Lab and then in the Genie project. That this steady march forward that, how these factors of two kind of form a very substantial product ultimately that really allows you to do completely different things. And then the community, right, invents all kinds of new ways of using these things. So these are just some of the things where kind of technology, sometimes in expected, sometimes in unexpected ways, sort of, couples to new insights and new kinds of biology.
Yeah, I always think it's amazing when one experiences like the rise of a new technology, which was how can I say, which is kind of a new thing at one time, but then it's so established later on, and it's kind of such a solid part in how science is done. And it's amazing to hear that you've been part of so many steps along the way over the last years. But yeah, with that, I actually would like to thank you again for participating in our seminar series. It was a pleasure to have you with us. But I think also because of the time, we will actually, I think I would say we will skip the meet the speaker session for now. I will just give a very brief outlook what you can actually experience or what you can expect for the next seminar. So the next seminar will actually be given by Ali Pasha Varisi, and he's from the Rockefeller University, and there he leads the Laboratory for Neurotechnology and Biophysics. And so his presentation will be already next week on February 1st. And again, we go back to the old time at 9 a.m. And this time, actually, it's so that seminar will be given here on campus. So that means Ali Pasha will actually come to the Geneva Research Campus and will meet a few group leaders here and other scientists in person. I hope we also managed to get the recording right. Fingers crossed. I will do my best. But otherwise, thank you very much again, everyone, for joining us, and see you all then next week already. Okay, bye-bye.